All right, welcome everybody. Happy Monday, happy time change. Man, all right. I just hope they do away with it. So, as we jump into some new stuff, I'm sure a number have already completed the exam, but just a friendly reminder, it closes today at 5 p.m. If you haven't yet done so, get on there and do it. All right. So we've talked about all sorts of different details. We want to think a little bit in this next chunk of material that is going to be cut in half by the spring break that's coming up next week. So we will go through a part of it and then get the next part of it. So 3 chapters that think about different ways that we might go about trying to deal with microbes, trying to control their growth. There are numerous things we might think about. We do some of this stuff in the lab. When you come into the lab and you use the disinfectant to clean the bench off before you start doing your stuff. And hopefully when you're done doing your stuff, you disinfect it as well. We talk about some of these steps that we might utilize to kind of ensure that the work we're doing is aseptic. We're being as clean as possible. And the microbes that you're working with are the microbes that we're giving you, and not something that you picked up off yourself or off the bench top or whatever else, right? So controlling growth is important, and it doesn't matter if we're talking in the lab or at your house or whatever it might be. There's definitely things that we might do to help ensure that things that we don't want to have microbes growing on aren't, like maybe slapping leftovers in the fridge, right? That could be a good idea, right? Maybe not, right? I think so. So this image, every time I look at it, it makes me think of your first day rolling into lab. Although you didn't get the opportunity to swab whatever the heck you wanted, I mean, sort of, but I think you kept it in the building. I don't think you had the opportunity to go out and maybe swab something in your car, but it sort of makes me wonder if you have a car and you did some swabbing, what sorts of stuff might you find, and would you want to know what's growing in your car? Probably not, right? There are all sorts of potentials when we think about options like this. And in this case, the interior of the car being an environment, and definitely something we wouldn't classify as being sterile, all right? So there's all sorts of different potentials. And many of us might say, well, I eat lunch in my car, right? There's all sorts of things that potentially get dirty, get touched with dirty hands, and potentially lending to areas that might be more rich for growth if there happens to be things that maybe got spilled, and provide a better source of nutrients for somebody that wants to try to live, right? So in this example, they're looking at all sorts of different touch points, things that you may or may not touch in your car that sets the stage for possibly coming in contact with stuff. So they did some swabbing, they did some streaking onto these plates, and an individual colony, right, is what we're looking at. Different types of colonies likely, and colony forming units are what we're looking at as we move through here and start evaluating all of these plates. How many colonies, individual bacteria that grew up countless times to give you a visible mound of growth, how many of those are we seeing? The more colonies, the more organisms we're truly thinking about. We also, and I know you swabbed a few things, that likely gave you numerous types of bacteria on there, and maybe even some fungus, right? And then there were probably other things that you did some swabbing with that looked like maybe one or two types of bacteria growing on it. So we could see the amount of colony forming units, but we also could see the diversity of those and get some ideas about what's truly going on. All right. So there's all sorts of things that they swab, and some touch points like the center console, which kind of makes sense, maybe a spot that gets more things potentially touched or spilled upon it, providing the opportunity for more things to maybe thrive. And other stuff, like the window control over here, not so much, all right? Door locks, other things, maybe not as much. Makes me wonder about your keys, right? And if you got the little key fob or something else, if you were swabbing that, what could you see? What about your cell phone, right? There's all sorts of things that I start thinking about with touch points and things that might have differing numbers of microbes as we look at it. So we've seen some images of this before. We've actually seen a couple of times with some of these BSL-4 as we think about controlling situations. Really kind of depends on your circumstances where you might be working in the first place. And some microbes, you truly want to ensure that they're kept where they ought to be, inside of that lab, inside of this hood, truly, all right, and not something that's allowed to get out. So the suit is a positive pressure suit that's piping air into it. The lab itself is negative pressure, so all of the air is being kept within there. If the door opens, air does not go out of the lab. Air from the hallway would be being sucked into this lab and blown out through the vents in this fume hood, right? The hood itself helps to provide that negative pressure, and everything that's being worked with in this should stay within this hood if everything's functioning properly, right? There are also levels of safety within these hoods, so we're not just sucking it up and piping it right outside. That wouldn't be wise. There are things in these hoods that have different potentials to control, like UV lights for some sterilization and HEPA filtration, that the air is going up and through these filters, kind of like a furnace filter, but hardcore, all right? Your furnace filters may be trapping some dust and maybe pollen and things like that. You might have some slightly better ones that might get some allergens, but the HEPA filters are more serious types of filtration that trap microbes, and virus as well can be trapped, dependent upon types of filtration, but typically BSL-4 stuff, you want to filter it all. So virus also gets trapped behind in these filtration mechanisms. So ensuring the safety of everybody, all right? So there's levels of classification when we think about dealing with microbes, whatever they might be. We've touched on a little of it, but we haven't actually really thought about the details. We've shown some BSL-4 stuff, the biosafety level 4, the highest risk, the potential, I think about the spacesuit wearers, as you're going to work, as you're leaving the house and telling a loved one goodbye, it truly could be, right? That could be the last time, right? That's a little bit disturbing when we think about it, right? These microbes are very dangerous, exotic, all sorts of things like, maybe Ebola and others that fit the stage of high levels of concern. Marburg is, both of these are viruses that are serious concerns, right? And cause some massive contagious situations with this, right? And they're things you, technically I would think not want to be working with. As we move down a step, Mycobacterium tuberculosis is our example here, is a BSL-3. These are things indigenous or exotic, definite potentials for some serious threats. And we think about, respiratory transmission as a likely mechanism for the spread of these things. So also warranting hood usage, but likely not something that we think about needing to don the spacesuits to ensure we're protected that way. But we're still working in a hood with this negative pressure idea to keep everything within there, but maybe not necessarily spacesuit, right? Maybe face shield, right? But little bit less of a concern as level 4, right? Still a concern, right? The level 2s and level 1s, these are things that are a little more easy to work with. We try with virtually everything in the lab that we have the opportunity to work with, we try to keep everybody at level 1. There are a couple organisms that technically get bumped up a little bit higher. The Staphylococcus aureus is one potential. Even though these bacteria have been attenuated, there are still some potential issues that they could cause some illness, all right? Not things that we truly think of as life-threatening, like up above, all right, but still some potential issues, all right? So we think of, varying levels of severity of disease that they may potentially harbor, and caution, definitely. I assume with all of these, it doesn't matter what level, you should be treating everything with high levels of concern and treat them as if they truly are disease-causing organisms. And if we all roll in there with that mentality, we're treating things carefully, we're watching ourselves and others to know that we don't potentially contaminate them, then everything should be all fine and good, right? So the level 1s, these aren't known to cause disease, or at least cause them in healthy hosts, or there's very minimal risk. So like some non-pathogenic strains of E. coli and other potentials, all right? I still think, even thinking, non-pathogenic E. coli, if it happens to get ingested, you're still going to have a little bit of intestinal discomfort, right? But not something that we truly think of as a serious disease causer, not like the, 0157 that gives you bloody diarrhea, all right? We're not thinking that, all right? But there's definitely some potentials, Salmonella or Shigella strains that could potentially cause, some discomfort dependent upon how those things are being dealt with in the first place. So a lot of stuff we're thinking about, like environmental, we will think a little bit about ourselves in the last 2 bits down here, but how do we deal with things to ensure, like I mentioned, your bench top, to ensure that we're coming into a clean work environment, right? So initially, the top 3 bits, we're thinking about inanimate objects, bench top, other things that non-living tissue that we may use some of these techniques. And it depends on what it is that we're dealing with as far as how we may use it or if we could use it. So disinfection, these techniques reduce or destroy the microbial load, again, on inanimate objects, all right? And we think about heat or antimicrobial chemicals, your cytocon, the bench disinfectant, and other substances, maybe household cleaning things, fit into this disinfection category, right? Bleach, phenols, Lysol, glutaraldehyde, all of these are disinfectants, right? Reducing or destroying, all right? And destruction oftentimes is what we would like. We would like to kill them, right? Sanitization, we're not really thinking about destruction in this category. So we're reducing a load to a level that we would classify as safe. There's still likely going to be some microbes present, but their numbers are very low, right? We also may, you know, be inhibiting growth, but you're not flat out stopping it altogether, slowing it, all right? So commercial dishwashing, they've got sanitizers within there, cleaning restrooms, sanitizers. I remember, you know, getting to swab everything. And my first thought, when I was taking some microbiology many years ago, my first thought of, oh, sweet, I can swab whatever I want. I'm going to go swab the urinal. That's going to be disgusting. And nothing grew on there at all. Absolutely nothing. All right. But I thought there should have been, right? That seems like a pretty dirty place. All right. But nothing grew. And then you back it up and you think right here, right? Sanitizing in restrooms, they use all sorts of things to ensure that they are clean. You hope they do at least, right? If the custodians are really doing their job, right? And in turn, it seemed effective, all right? So handful of different potentials with this that could be utilized. Sterilization, this is definitely more of an extreme idea. And if we're thinking about like a surgical procedure, you would hope those instruments have been sterilized, right? If you're going in to get a tattoo, you would hope that those needles have been sterilized, right? You would hope that you could look around and maybe see a little benchtop autoclave and see that the materials they're going to use on you are coming right out of this autoclave. And the tattoo artist doesn't just wipe it off on his pants and start going to town on you, right? If you're getting a tattoo in a basement, better think about that a little bit, all right? Back alley tattoos, all right? Not so good. All right. Autoclaves are a great example of our sterilization. Pressurized steam, we could use some chemicals or radiation as potentials for sterilization as well. Things that may not tolerate the high heat or pressure, we may employ chemical or radiation methods, all right? The Petri dishes that you guys work with in the labs come in a sleeve of 25, and they're all in a big plastic sleeve. Those Petri dishes are sterile before they get pulled out of the sleeve. The media gets sterilized in an autoclave. It gets cooked. It gets cooled to handleable temperatures, about 50 degrees Celsius, which is still pretty hot, all right? But then they carefully pour it into a sterile plate. And if they've done everything properly, those plates shouldn't be contaminated. Sometimes lifting the lid off and pouring stuff in there, a little bit of microbes do end up contaminating it. We see some of that stuff happening, all right? But when they first come out of the package, they've been sterilized, and they've been sterilized with some chemicals, because the plastic Petri dishes can't get sterilized in the autoclave, they would melt, right? So they use chemicals, and we'll touch on some of that stuff in a little bit. So all of these, we're thinking inanimate stuff, all right? I'm going to give you one quick little story about sanitization and sterilization, because they're not the same, all right? This reduces and this completely kills, all right? So I'm a microbiologist. I have a yard that has a handful of fruit trees, which in turn, you know, if life gives you lemons, you make lemonade, they say, right? Life gives you apples, you make hard cider, right? You must make alcohol, right? Is really how I feel, right? I've got all this fruit, I must ferment it, right? Seems like a good idea, all right? So in doing this stuff, there are different ways you might be treating the bottles that you would be putting your product into, all right? You would hope they've been washed, and you would hope that they've been cleansed in a fashion that would make it so there isn't any fouling, any bad bacteria or something in there to make what you've produced not be tasty, right? So oftentimes they get sanitized. in an iodine solution, and then you can put your alcohol in there and you're good to go, all right? And oftentimes the alcohol's enough that as it goes into this bottle that's been sanitized, you're good to go, all right? That's kind of the key, alcohol, right? Going in there, then you're okay, all right? So I had all of these apples. I was juicing all of these things, and my wife and children thought maybe instead of turning it all into some alcohol, maybe we could just bottle some of this up as apple juice, because wouldn't that be tasty too? And I agree, right? Maybe a little apple juice would be all right, all right? I had already juiced 5 gallons worth of apple juice. That's a lot of juice, all right? And I had all of these wine bottles that had been sanitized. They hadn't been sterilized. They'd been sanitized. That's the key, right? That's my mistake. As I put the juice into these bottles and I cork them all up, now I'm thinking we're good to go, right? This apple juice is in these bottles. They've been cleaned, right? I've soaked them in iodine. I let them air dry, right? There were still, in a few of those bottles, there were still a few residual yeasts that were available. And once they got sugar, once they got the apple juice put in there, those yeasts woke up and they started growing and they started fermenting and they started turning that apple juice into alcohol very slowly, all right? I had these bottles in my basement and I walked downstairs and there's a puddle on the floor. And I'm like, well, this is weird, right? There is nothing that should be leaking. I'm looking at the ceiling. I'm wondering what's happening here. How is the floor wet? And then I look over and see the bottles, and one of them has blown a cork out and all of its contents have kind of spewed out onto the floor. Well, that's a problem, right? That's a mess. And then it hits me, right? These things, they're fermenting. What have I done, right? I didn't sterilize, right? So a few of those yeasts in a few of those bottles started going to town. And as things start to grow, you've seen some of these things in some broths that you've got. It goes from clear to turbid, right? So I could see some of my bottles that looked nice and clear and some other bottles that were a little bit turbid. And I knew those turbid ones, they're not apple juice, all right? They're not for the kiddos, all right? That's bad news, right? Those are for dad, right? And in turn, the first bottle that blew its cork out, I smelled it and I'm like, oh, I could smell a little alcohol. Didn't smell bad. So being a scientist, obviously I have to taste it to see what's up. And I'm like, oh, a little bit of fizz, a little bit of apple deliciousness, tasty, right? So I put a little in a glass, handed it to my wife. She tasted it and she's like, what? You made that? That tastes good, right? That hurt, right? That cuts like a knife, right? Because when I'm making stuff for real, if you can make it the most concentrated alcohol you can without needing to use a still, why would you not, right? It just seems like that's what you ought to do. This was very low potency, all right? Very low, like Utah beers, all right? Low, all right? But tasty, all right? And in turn, those cloudy bottles, well, I consumed them, all right? The other ones, technically, they were cleaned more thoroughly, I assume. So then it goes back on me as well. I obviously didn't wash those bottles very well if there were a few yeast cells in there. But then we flip it back around, and if that apple juice had been turned to alcohol when it went into those sanitized bottles, it wouldn't have been a problem in the first place, right? Nothing would have mattered, all right? If it was bacteria, it'd be kind of a different story, but that's not really what was up, all right? Sterilizing gets rid of all of that, all right? So my next round of juice, I decided I'm gonna just truck all these bottles up to the biology building and throw them all in the autoclave and cook them up, all right? Then they're sterile, and then I'm good to go, all right? I could also do some sterilizing at my house, but in the heat of the summer, after I had done it a few times, and then my wife learns I could do it elsewhere, the elsewhere won out, all right? You can sterilize bottles in an oven by cooking them at 400 degrees Fahrenheit for 4 hours, right? That's super hot. And if your house is already hot, and now you're baking stuff at that temperature for 4 hours, people aren't very happy about it, right? And in turn, the autoclave can do that in about an hour, all right? And it doesn't heat up my house. So that's my story, right? Sanitizing and sterilizing, they're not the same thing, right? And truly, if you're trying to put just some juice in a sanitized bottle, maybe setting yourself up for some problems, all right? People will make beer and they add a little bit of malt, the stuff that's the sugary stuff you're gonna be fermenting in the first place. You can add a little malt to your fermented beer and bottle it up, and then it will carbonate all of those bottles. They also could put a little bit of sugar in there and do the same thing, all right? If they don't do the right amount, they put too much in there, when they cap those bottles, they carbonate and then they blow up, all right? And that also could be a huge mess, all right? Never had that happen, just the apple juice fermenting on me. Okay, enough of that. So when we think about ourselves, we're not thinking about the top three, all right? You definitely don't want to do any of that. You don't want to autoclave yourself, right? And any of these other techniques, truly probably something you really wouldn't want to get on your skin in the first place. The disinfectants, the cytochrome that we use in the lab, we dilute the crap out of that stuff to make it so you can use it in the first place. We buy it in a gallon jug, but we dilute it in 5 gallons of water with 1 ounce of the cytochrome. A small little cup, all right, goes in to 5 gallons, and that makes it a concentration that's doing its job. If you get it straight out of the bottle and it hits your skin, it burns, all right? I know that because I felt it, all right? Got a little blister as it touched my thumb. Then I knew, right? That'll teach you, right? So you want to be cautious with those things. Antisepsis and de-germing are things we think about for living tissue. So in both instances, reducing the load, but we're not eliminating, all right? We're cleaning stuff off, right? So antiseptics reduce the load, and we think about some sort of antimicrobial chemical that's put on there, all right? Boric acid, isopropyl alcohol, peroxide, and iodine, back up here to the sanitization, right? Can be used for some of that stuff. It's also used on your skin. If we think about prepping for surgery, betadine's a good one for that, all right? De-germing would be cleansing. You know, you get a wound or something like that, and you need to get stuff out of there. So de-germing it is scrubbing, gentle to firm, dependent upon what's up, all right, to get rid of stuff. Hand washing is another example of some de-germing, right? Soap, alcohol swabs, both potentials for this mechanism, right? So really depends on what you're working with as far as how effective it might be. So we saw before growth curves, and we saw bacteria hanging out as they first get inoculated in that lag phase, and then we see some exponential and stationary, and then we start to decline. the death phase, right? As they've used up nutrients, right? Well, this is that, but it's a little bit maybe more extreme. This is thinking about looking at those respective processes we just showed and thinking about maybe how effective they may be. So really the inanimate objects might be more of the idea with this. So how is it, how effective is it for the destruction of those microbes? And we can see massive amounts of cells and bringing those numbers down. We still have numbers, right? So we could see it dropping very rapidly, depends on what we're looking at, all right? So how much how much time it takes to kill 90% of a population, that's a single log decrease as we move through here, right? With whatever agent we're looking at, all right? So that's this one little square, right? The D value, how much it takes to go from this to that. And it could be from a few minutes to pushing an hour. It truly depends on some of these circumstances as far as what you might be needing, all right? The disinfectant you put onto the bench top truly should be working by evaporation. You should put it on there. It should remain sort of wet for about 10 minutes till it dries to truly be effective. And oftentimes as you roll into the lab, you spread it on there and you probably wipe it dry, right? But I hope at the end of the lab, you spread it on there and you leave it a little more wet. You're still going to be wiping it, but leave it a little more wet so that it does air dry. And then the next players coming in have more of a clean environment. They can wipe it dry and we keep going like that. And as long as everybody's kind of doing the same thing, we hope everybody's doing the same thing, you come into a clean environment. Even though you still disinfect it, it should be clean as you're rolling in there, right? So these are really the same idea, just different ways of doing stuff. So this is a Bunsen burner, which we all work with in the lab to sterilize stuff, flaming the loop. So heat, right, as a mechanism. As you flame the loop, any microbes that were on there get incinerated. They get cooked to a crisp. The hopes are that you're, sterilizing all of this stuff properly, so not exposing yourself to like aerosolized bacteria as we see the tip of the loop going in, and it all gets kind of cherry red very quick as the heat's hitting it, right? Right up above the little blue cone is where you see the hottest part of that flame, all right? So this is a very common technique for sterilizing stuff. We use the Bunsen burner to heat fix bacteria to the slides. We use it for lots of things, all right? Also just doing your work in the close proximity of that Bunsen burner. The hot air helps to keep microbes away from settling back down into your whatever, your stuff you're working with, your plates or your tubes. It keeps it kind of a sterile little environment right around that Bunsen burner, all right? You shouldn't have your fire going over here and you're doing all of your stuff plating, clear away from the Bunsen burner. You should be close to the flame, all right? Should be, all right? This is an incinerator, all right? Bacterial incinerator, whatever, it's an incinerator. It's the same idea as our Bunsen burner. It's just a little bit different, all right? And it's got a big kind of ceramic core going on the inside. You can see it right here, in white. A screen around it to keep you from touching it. Even if you touch this, it's going to burn, right? And there's a heating coil that's going around that ceramic core, kind of like a toaster is really what this is sort of like, all right? And you can see these little heating elements getting red hot, kind of like our inoculating loop right here, right? And inside of this, it's like a little furnace. It gets super hot, but it's all enclosed. So if you were working with things that were real pathogens, First off, this would probably be in a hood, right? You'd have it in there. It would be on glowing red hot, and your loop goes in this chamber. And anything that's on that loop that's potentially going to get aerosolized stays in that chamber and it gets cooked up. So it helps to prevent any sort of agents from maybe getting inhaled, right? Kind of just depends, right? And as I look at this, it seems almost as if this picture was taken right in the biology department in somebody's lab. There's crap all over the bench. It makes you wonder, couldn't you clear it up a little bit? And then I back it up and I'm like, but this is really kind of showing real science, right? This is what it's sort of like. We've got our pipette box, we've got a beaker, we've got some filter paper and lens paper, all sorts of things on this bench. And like I said, it kind of looks like you could be rolling into a handful of people's labs in biology and see this kind of setup, even though we're taking a picture for a book, right? Comb your hair a little, right? Nah, forget about it, right? Mad scientist. So autoclaves, it truly depends on where you're working at. I mentioned the tattoo shop. Might have just a little teeny benchtop job that gets a handful of things put in there, right? Or you may step it up a little bigger, and you could be autoclaving flasks of media. In this case, this autoclave, you're seeing a kind of cross section of what's up in this, a big chamber that you could wheel stuff in or out and have lots of bottles that are getting autoclaved. You could have numerous wine bottles going into this thing to get sterilized, right? All sorts of things might be able to be sterilized in a larger autoclave. There are other situations. I would like to imagine that the real autoclave is right here, right? Behind this huge stainless steel door. In some situations, like maybe a hospital situation, it could be more like this, where you've got two rooms. One room is the dirty room, and the other room would be the clean room. And that room is separated by a gigantic autoclave. And only stuff that needs to be sterilized, it goes in the dirty room. And on the other side, in the clean room, that door opens up and everything coming out of the autoclave is now sterile. It would make sense that you would not want to be autoclaving stuff and then pulling it out where there's a bunch of things that could potentially be contaminating what you just autoclaved, right? So by keeping that separate, dirty stuff on one side and clean stuff on the other, then there's no chance that the clean stuff comes in contact with the dirty or vice versa, all right? A different situation. And there definitely are some autoclaves that are gigantic room size that you could walk right through, and you could be cooking numerous things, maybe wheeling carts in there and those carts sterilized with everything else that's on it, all right? Kind of crazy, all right? Not the same idea, but sort of similar. In biology, the animal facility where we work with some rats and mice, rabbits, and some other things, the cages that some of these guys are living in, well, they need to get washed. And there is a huge cage washer that is kind of like I just described, a room size dishwasher, right? Cage washer. You could walk through it. And there's two rooms, the dirty room, and the other side where it opens up is the clean room, and no dirty stuff in the clean room, right? It all ensures that those cages go from one side to the other, and then they're back into use. Water bottles and all of those things that the animals need go through those cage washers and get cleaned, right? Crazy, right? And there's a little safety pull thing. So if you happened to accidentally be in that cage washer and it got closed and turned on, you could stop it and then get out. And if you didn't, I'm pretty sure you wouldn't be alive when it was done cage washing. wash the crap out of myself. All right. That's crazy. So in an autoclave, there's a couple of ways we could see if it actually did what we thought it should do. These tubes have some autoclave tape on them. You've probably seen some stuff in the lab with some black stripes on it. And the black stripes on this tape is indicating that it has gone through the autoclave and that autoclave came to the proper temperature to actually do its job. It changed colors. And if it didn't reach the right temperature, it would come out and it would look like it did before it ever went in, just kind of like a piece of masking tape. You can see sort of the white stripe in there. But if it hits the right temperature, then you see this black mark, right? Knowing that it's been sterilized. There's some other ways they can do it, little ampules that have a bacterium, Clostridium, in there. Clostridium is a spore former. If you put that in the autoclave and then you pull all your stuff out and you crack that ampule, it inoculates it right in this little tube that it's in. And if it killed it, it does nothing. But if that bacteria is still viable, if those spores are able to hit that media and start growing, it changes colors. And then you know everything that you thought was sterile technically is not, all right? Autoclaves can get overpacked. They could put too many things in there or have them stacked up upon one another. And that sets the stage for maybe something in the middle that didn't actually reach the right temperature. So knowing it truly all got cooked properly is important, all right? So there's some other details when we think about some of this. Thermal death points and thermal death times. The death point is the lowest temperature used where all of the microbes are killed in 10 minutes, all right? So what temperature, what's the lowest temperature I can use to kill everybody, all right, in 10 minutes? The thermal death time, same but different. Now it's the length of time needed to kill all of the microorganisms in that sample at a given temperature. So now it may be less than 10 minutes. It may be an hour and a half, all right? Just dependent upon how you're trying to actually do it, all right? To make it effective, right? Like baking the wine bottles at 400 degrees Fahrenheit for 4 hours should be effective to kill off anything that wanted to live in there, all right? So we've talked a little bit about Louis Pasteur. We said a handful of details about some of the stuff he did. He did more than just pasteurize our milk, but this is where we want to think about a little bit, all right? Pasteurization initially was created not for milk, but for frothy beverages, for wine, right? And beer, but those that we don't want bacteria to get in there and make it not taste good anymore, right? Does it actually taste good? That depends on the person, right? So pasteurizing it helped to keep it fresh, right? So it lasts longer. You can maybe transport it longer distances and things will stay stable, if you will. So then we take it a step further and think, well, geez, we ought to use that on milk, right? There's all sorts of potentials with this. And there are numerous organisms that we can see in raw milk that set the stage for some potential ill effects, all right? So through the process of pasteurization, the hopes are that we eliminate all of these pathogens. There are still, dependent upon pasteurization techniques, there are still some organisms that might be present in that milk, but they would be more thermophilic bacteria. And those higher heat-loving bacteria, we don't really think about those guys being pathogenic, right? So not so much of a concern. Some of those might be good candidates for the production of yogurt. A lot of them tolerate a little bit higher temperature and in turn can ferment the milk when it's cooled down to the proper temperature and turn it into yogurt. So these two pasteurization techniques, the first one, more common, heating it up to a steam to about 72 degrees Celsius, and it's kept at that temperature for about 15 seconds. Then it's bottled, refrigerated, good to go. All right. Once you crack that lid open, technically the potential for stuff to get in there and for it to spoil truly is there. Our next one, this is a high heat. So we are bringing it up above the boiling point, 138 degrees Celsius, held there for just a fraction of the time, 2 seconds or maybe a little bit more, but just a couple seconds, all right? And then it's quickly sealed in containers, and it could be stored on the shelf for months, upwards of 90 days without any refrigeration. There are no microbes present in this milk, right? It's got a shelf life, not a refrigerator life, much longer than regular pasteurized milk, all right? And in turn, it's also, I'm not a milk drinker, but it's got a different sort of taste, right? The high heat pasteurization versus the regular does a little bit to some of the sugars in the milk, making it a little bit sweeter. So I've been told, right? It also impacts some of the protein as well that's in there, all right? But a little different, all right? There are other areas, some other countries, European countries that use some radiation with the milk, and it in turn, same idea, shelf life, right? Can be on the shelf. And again, no need for refrigeration till you open it. Once you've opened it, it doesn't matter if we've irradiated or high heat pasteurization. Once you've opened it, it truly needs to be refrigerated, right? So boatloads of different bacteria. Campylobacter was one of the main ones. It's a nasty diarrheal disease. Coxiella, Listeria, the bad E. coli, TB, another cousin of it, paratuberculosis, some salmonella, and Yersinia, cousin of the plague, enterocolitica. All of these bacteria that we would think of as, potentially being normal flora in the cows, and coming out the back end of the cow, potentially contaminating the udders. And as the cow is milked, the milk's contaminated as well, right? So pasteurization helps to eliminate all of those possibilities. So we'll look at this and then call it a day. This bit right here is thinking about preserving stuff. So some bacteria or other specimens, doesn't have to just be bacteria, there are some things that we would like to preserve long-term, maybe indefinitely, and putting them into an ultra freezer. This might look like a big chest freezer that maybe your parents have in their basement or out in the garage, right? Keeping all of the meat and stuff frozen. But this is a little bit different, all right? This freezer goes super cold, -70 or -80 degrees Celsius. That is ultra cold, all right? You don't just take bacteria and put it right into the freezer. It needs to be put into a glycerol solution, which is a like super thick kind of syrupy solution that helps to keep those bacterial cells from exploding as they get into these cold temperatures, and they stay viable. You can open them up, grab a little bacteria out and pop it back into the freezer, and it stays good as long as you're handling things properly, all right? So we see here this freezer, this ultra freezer getting opened, and there are 3 like chambers in this freezer, each of which covered with a chunk of styrofoam that keeps it so that the other parts of the freezer aren't getting exposed. And this lid is open, you grab your stuff out and you close it very rapidly. And as you see here, this guy is wearing super thick gloves, right? You wouldn't touch stuff in here with bare hands, right? You've all seen Christmas Story, right? Licking the flagpole, right? You don't have to lick anything, just your bare hand touching. Inside of here, lots of things are stored in some metal containers, which seems kind of dumb. Put a bunch of metal inside of a super cold freezer and then touch it, right? your hand would stick right to that. That's why you wear these thick gloves. You grab it out, get your stuff, and slap it back in here. This last bit right here, this is liquid nitrogen. Maybe you've seen some stuff with liquid nitrogen, the banana getting dipped in liquid nitrogen and shattering, or a rose going in there and it just blows apart. That's pretty cool stuff. This is super cold. And we think about stuff, we call it snap freezing. It's basically frozen instantaneously. As soon as that liquid nitrogen hits it, it's frozen solid, right? And so a different mechanism for preserving things. All right, I will quit with that. Farewell.